Hello, everyone. Welcome. My name is Liming, and thanks for having me at the Aykut Kence Evolutionary Conference. Today, I would talk about my research on parasitic plants. Now, give me a moment to share my screen. And here it is. What makes a parasitic plant? So first of all, what it is a parasitic plant? Parasitic plants are a group of very special plants that gain some or all of their carbon, nutrient, and water from other plants, namely their host. For example, here we're seeing a thread-like plant called dodder. Its Latin name or scientific name is Cascuda. It sort of wraps around its host and also bite into its host through a root-like structure shown here on the right that is called hostoria. So hostoria is really a special, highly modified organ that the parasite use to make or establish a physical connection with the host, and then it usually penetrates into the xylem or phylum of the host, and then they extract everything they can get from the host. Across the flowering plant tree of life, especially in flowering plants, parasitism has evolved 12 times, which are highlighted by these red lineages. And then in total, there are more than 4,500 plants that are parasitic. Within gymnosperms, parasitism is highly rare, but we do have one genus that has also become parasitic, which is the Parasitaxis. Which is a sort of a woody, shrubby plant that feed off other plants as well. And then in terms of growth form, parasitic plants really have an exceptional diversity. Oftentimes we have root parasites, such as the Rafflesia, which produce the largest flower in the world. And also things like Cistanchi or Striga, who basically establish connections with the root of the host and get the water and nutrient they need from the host. Other than that, we also have stem parasites, such as the mistletoes and the dodder, that we, and just to talk about that before, these parasites, they attach to the stem of the host, the photosynthetic part of the host, and then also get the all of their needs from these stem-based connections. Another way to classify parasites is based on whether they are photosynthetic or not. So here we have hemiparasites versus holoparasites. So hemiparasites, despite being a parasite, they are still green and can conduct photosynthesis. Examples would include things like the Indian paintbrush, in the Orancaceae or the mistletoes, and then always, or as well as a handful of other families in general. And then on the other hand, for holoparasite, they have completely lost their ability to conduct photosynthesis. And then within these various groups, we usually observe the highest level of morphological and physiological modifications. And then another thing I want to point out here is that within hemiparasite, there are also facultative and the obligate parasite. This means that facultative parasites, they can complete their life cycle without a host, while obligate parasite, they require to be attached or associated with the host in order to complete their life cycle, like flowering and then producing fruits, etc. The overall speaking, the evolution of parasitism, it follows Dollo's law. Basically, here's a texted description, but basically what Dollo's law means that this is a one-way ticket. There's no going back. Here, the Dollo's law, it says an organism would never return exactly to a formal state, even if it finds itself placed in a condition that was exactly as the ancestral state. Basically, what that means that once a plant becomes a parasite, it cannot go back to be free living again. And then the best example to demonstrate this this law or this principle is within the parasitic family Orobanchaceae. And then Orobanchaceae is a really exceptionally diverse family compared to other parasitic lineages that contains more than 1,000 species that's distributed worldwide. And the basal lineage is a free-living Lindenbergia. And then as you sort of go towards the more recently diverged lineage, you will get facultative parasites and obligate hemiparasites, and then you also get multiple independent loss of photosynthesis abilities to create different lineages of holo parasites. So they really, as you go from the basal lineage to the more recently diverged lineage, you have these nice sequential events of reduced photosynthetic ability, increased dependence on the host, as well as more and more decreased to absent vegetative tissues, including leaves and roots, and then also greatly modified body plans. And then one interesting phenomenon within plant parasitism is that hyperparasites, they do exist. So first of all, when we think about hyperparasite, we usually think about the relationship between, for example, a parasitic wasp that would parasite a butterfly caterpillar, and then there is a hyperparasite, another wasp that can parasite these parasitic wasps. So they form a tritrophic level relationship. And then the similar level of complicated multi-trophic level relationship can also apply to parasitic plants as well. So here we are looking at a really fascinating example from the field. On the left, we have the host plant. I think this is a monocot, maybe from the Bromeliaceae or Bromeliads. And then on the right, we have, seems like a mistletoe that parasite these monocots. And then on top of these mistletoe, we have these strenuous plants that really are the daughter, the coscutas, that are actually hyperparasites that feed off these parasitic mistletoes. So this is a really fascinating example of how the multi-trophic level parasitism can become quite complicated and interesting for ecological studies. So now to, after sort of talk about the background information about, on plant parasitism, I want to begin my talk by introduce you to several lineages of, in my opinion, really awesome, amazing, unusual parasitic plants. And then I will talk about sort of the scientific discoveries behind these parasites and how they, each of them as a model system, advances our understanding of plant parasitism as a whole. So first of all, Striga. Striga, the common name is witchweed. It's a genus that's in the Orobanchaceae family. And then the plant is shown here. They are these really beautiful purple plants with green leaves. That means these plants, they are hemiparasitic. They can do photosynthesis. And then when we place it on this spectrum of the degree of parasitism, we would like to place it on the left-hand side because it represents a lower level of parasitism. That is because it can conduct its own photosynthesis. So it's not as reliant on the host as other plants. However, the common name of witchweed really comes from the sense that they are a devastating and aggressive crop pests. Each year, they cause over 10 billion US dollars of losses in Africa alone because they primarily target at corns and rice and other monocot crops. You can see a photo from the field that once agricultural land is infected by these parasites, the reduction in the yield is really significant. Sometimes there's no yield at all because they have taken up all of the nutrient from the host. And then they have been estimated that 100 million people in West Africa have been affected by this pest. And then because they are so sort of nasty in terms of their passive nature, a lot of studies have been focused on how they establish, first of all, how they recognize their host. And then I thought this is absolutely fascinating. If we look at the figure, let's focus on the left side. So for a lot of plants, including corns, it will release a hormone called strigolactone to attract microrhizobial symbionts, to attract basically fungus and other beneficial microorganisms to the rooting areas. And then what Striga does is that it recognizes this chemical cue and use it as a way to physically locate the root of the host. And then this will trigger the germination of the seeds, and then the seed will grow towards the direction where the chemical cue is released. And then sort of the parasite would develop and establish haustoria and also taken away all the nutrients, so that we can see on the right, it causes really devastating results for the host, while the parasite itself, it definitely thrives by these parasitic relationship. And then a second parasite I want to introduce here is the daughter. The scientific name is Cuscuta. It's a genus within the Convolvulaceae, the morning glory family. And then because Cuscuta, it's at the interface of hemi to holo parasites, meaning that some species within this genus can do photosynthesis, while there are also other species within the genus that has lost their photosynthetic ability. And then I think there are quite a few Cuscuta that's distributed in Turkey as well. So if you have this search image, they form a really massive network or web-like structure when they parasite a certain host. But themselves, they are really delicate looking. These are basically strings with hostaria, which we talked about before, that penetrate into the host. And then on this spectrum of a parasitism, we will likely place it on sort of some of the middle point, where it represents a transition place from a lower level of host dependence to a higher level of host dependence. This Cuscuta, because it was very easy to be cultivated in a lab, and it has been used as a model system to study plant parasitism at molecular level. And then one thing we learned about Cuscuta and its relationship with the host is that, both DNA, RNA, and protein molecules are frequently exchanged between the host and the parasite in the hostarium. So first of all, there are 2 points here. This is a two-way traffic. It's not unidirectional. And then the second point is that there are both these genetic materials as well as small to big chemical molecules. Many of them are secondary metabolisms that has been frequently exchanged or trafficked between host and parasite. Then as a result, it really leads to a lot of fascinating phenomena. And then one example I really liked is that because we know that flowering plants, they flower at different time of the day and different season of the year. And then the flowering is regulated by environmental cues, for example, the day length. And then below is a chart of what kind of a flower you can get in each time of the year. However, as a parasite, you might need to think about it because you are reliant on your host, and then an environmental cue may not be as beneficial as the cue that's directly from the host. In other words, it might be beneficial to flower as your hosts do, because both of you can attract pollinators, etc., or you can exploit all of the exceptional nutrient or energy that your host is producing during this reproductive period. And so as a result, first of all, the flowering locus T, which is FT for short, is one of the important proteins that has been identified by scientists to activate flowering in many systems, such as the Arabidopsis, and then in some monocots as well. So it's a regulatory protein that receives the signal from some light sensing proteins, and then it will also transit this signal to activate all of those complex programs that plants have to begin the reproductive phase. And one thing we found interesting about Cuscuta is that the FT gene, this important, essential flowering gene, is lost in the parasite. As well, as a result, it has to rely on the FT protein produced by the host to regulate its own flowering time. And then to demonstrate that, researchers have planted Cuscuta, the parasite, with 3 different hosts that flower at the different time of the day. These would include the wild tomato, tobacco, as well as cucumber. So the x-axis is the host and 3 pairs of a host parasite. And the y-axis represent different time of the day. For example, wild tomato, it flowers earlier in the day, and then the tobacco flowers at different time, and cucumber flowers even later. And then what's fascinating is that within each pair, the parasite daughter, it flowers exactly at the same time as the host. And then this directly provides direct support for our hypothesis that parasite, it not only uses the nutrient it gets from the host, but also uses the various types of chemicals from the host. And here it used the flowering signal from the host to activate its own flowering. And then a fourth, and then a really weird type of parasites that I want to introduce here is Hypnora, excuse me. And then at the first glance, it really is a weird looking plant. You may not even associate it with the plants given its appearance. This is a plant that you can find in a desert, dry areas in Africa, both South Africa and Namibia. And then it has completely lost its ability to conduct a photosynthesis. It's deeply dependent on its host. So on a spectrum of parasitism, we would like to place it towards the right side, representing a higher level of parasitism. And then just by looking at it, you can use all kind of your imagination. It definitely inspired artists from time to time to create what we think unusual or even alien organisms. And then the reason that it has such a weird morphological character is indeed, I believe, because of its higher level of parasitism. So first of all, the reproductive strategy Hypnora has taken is also very unusual as well. Unlike other plants who like to produce beautiful flowers to attract the bees or butterflies, it produces a weird looking flower that smells like rotten flesh. And then this plant is also hypothesized to be thermogenic, meaning that it can produce heat to keep itself warm. And then the really foul odor that it releases during the reproductive phase is to trap carrion flies or beetles for pollination. Here we have a very nice image of that. Here showing some beetles trapped in the flower and is covered in pollen. And then panel C is a enlarged image of the pollen that was stick to the elytra of the beetles. And then when I talk about Hydnora, it might have reminded you of many features of Rafflesiaceae, which is the plant that I'm going to focus on today. Rafflesiaceae, many of you might have heard of it because it produced the largest flowers in the world. That's the species Rafflesia arnoldii. And then it is even more striking that it's not supported by any visible or identifiable vegetative organs, including leaves or roots. And then, as we shown here, that there are 3 distinct genera within Rafflesiaceae. All of them, like Hydnora, it will produce a really unpleasant smell when it flowers to attract carrion flies for pollination. This is a really old lineage. It was hypothesized that the family originated in the early to mid-Cretaceous, and then there's a long sort of evolutionary stable status, and then they do not diverge until recently in the Miocene within each of the 3 genera. When we take a closer look at the different stage of its life cycle, it started as a mysterious seed, and nobody quite understand how they penetrate into its host. So one thing that's really special of Rafflesiaceae is that they live entirely inside their host before they emerge as a flower. So that means that they exist as these strands of cells, almost like a fungus mycelium within the host for an unknown period of time. It can be as long as even 5 to 8 years before it produces a bud that will eventually penetrate the surface of the host and develop it into a huge flower. And then again, like Hydnora, the flowers produce a really unpleasant smell to attract carrion flies for pollination. We could see that even see here, there are carrion flies laying eggs on the flower. So thinking that they might found a corpse of a rodent. It was actually hypothesized that these hairs and these filamentous petals will mimic the hair and the tail of a rodent. So that at least based on the photo, you can see that it's very successful in terms of its deceptive morphological mimicry to a to the fly, and then they definitely rely on the fly for pollination. And then it was also hypothesized that their seed dispersal is somewhat related to ants carrying them around. One thing I want to highlight here is that, again, for Rafflesiaceae, it's also connected with the host through this structure called hostorium. And then both DNA and RNA are frequently exchanged between the host and parasite. As a result, it creates opportunity for horizontal gene transfer, meaning that the genes from the host will be transferred to the parasite and fixed in the genome of the parasite. And then the sort of the gold standard to identify horizontal gene transfer is to use phylogeny, namely evolutionary gene trees, to identify such genes. So for a normal vertically inherited gene, we would expect the parasite to cluster with its close relatives rather than with a host. However, in case of horizontal gene transfer, because the gene is inherited or transferred from the host, we would expect the parasite to cluster with the host. And that in the case of Rafflesiaceae and its host in the grapevine family, because these two species, they are well separated from each other by more than 100 million years, it's very easy to pick up such phylogenetic signals. And then this phenomenon has been recognized by scientists about more than 10 years ago. But one remaining question or mystery is that the parasites are older than the host. So here we are looking at the age estimation of the parasite on the top and the host lineage in the lower part. As we can see here, there's a sort of an age gap between the parasite and host. And then the most parsimony explanation would be that the parasite, the ancestor of the parasite, was associated with a different lineage, a different host lineage in the past. And then to demonstrate that, we can again use horizontal gene transfer. Usually characterize extinct host parasite association are extremely difficult, especially in the absence of fossil evidence. But here, horizontal gene transfer, it really provides a unique opportunity because it can serve as a DNA fossil. So let's assume that a parasite has been associated with 3 different hosts in the past. Because DNA are constantly transferred from the parasite to the host, from the host to the parasite. Today, by sequencing the genome of the parasite, we can identify this DNA remnants from different hosts. And by doing a phylogenetic reconstruction of these horizontally transferred genes, we can then locate the affinity, the phylogenetic affinity of these hosts. So to do that, we have gathered genetic information from all 3 genera within Rafflesiaceae, the parasite lineage, as well as all representatives within the host family, the grapevine family in Vitaceae. And then we reconstructed a phylogenetic tree for each of the horizontally transferred genes identified. And what we found is that first, for all of the recent gene transfers that was received by crown group Rafflesiaceae, the host or the donors of these transferred can be traced back to tetrastigma, which is exactly their current hosts right now. However, for more ancient gene transfers that was shared by all 3 genera within Rafflesiaceae, That means these gene transfer took place in the common ancestor of Rafflesiaceae, so it is older. And then when we look at the donor lineages, we find a much more diverse distribution of the host. And then within them, we especially found that the Ampelopsis, or a close relative of Ampelopsis, it was frequently identified as a potential donor of these ancient gene transfers. So one possible explanation from this phenomenon is that prior to the association between Rafflesiaceae and its current modern host, Tetrastigma, the ancestor of Rafflesiaceae actually is associated or parasites Ampelopsis, or its close relative indeed. So with that, I would like to thank my advisor, because this is my PhD thesis work, as well as my co-authors. I received a lot of support from the Harvard OEB department, the Harvard Bioinformatics group, as well as professional societies to give me opportunity to travel and present. And then I also want to acknowledge the indigenous peoples of Malaysia and Thailand, on whose traditional territories we collected our plant materials. And thank you all for listening.